for taking time to participate and to sharing a little bit about his thought processes and how someone comes to terms with having a rare disorder that can be diagnosed even though there's no specific treatment yet for it, an experience that I think he describes quite well. I want to start by thanking people ahead of time, just in case we run out of time. In particular, I want to call out Chris Ross, who intellectually drove much of the work that I'm going to describe, and Susan Holmes, now on our adjunct faculty, who cloned the Huntington's disease-like 2 gene. There's a long list of people who participated in the laboratory and clinical efforts here. I'm sure I'm missing some. And of course, our funding sources, including the ABCD Charitable Trust and the Abramson Fund, and a special thanks to the support and assistance of the many individuals in HD, HDL2, or schizophrenia and related disorders who've participated in our projects and provided the inspiration. So I'm going to talk a bit about Huntington's disease, and then more about Huntington's disease-like 2, including its similarities to Huntington, the complexity of its mutation, and an interesting aspect of genetic diversity that HDL2 reveals. And each one of these, I think, will have some relevance to broader psychiatric disorders as well. So let's begin with Huntington's disease. This fellow up here is George Huntington, who described Huntington's disease when he was a 22-year-old brand new physician, never published anything else again. He described this disease in a way that still works. It's really a remarkable description. It's a disease that begins typically in middle age, but with some variability. It includes a progressive movement disorder with chorea, brief jerky motions, and dystonia, more sustained twisting movements, incoordination and ataxia, later rigidity and bradykinesia, along with a swallowing disorder. There's progressive cognitive loss, including early a subcortical pattern of dementia, which affects, for example, executive function, and later a global dimension. Psychiatric symptoms are common, including mood disorders, apathy, rigidity, irritability, and personality changes. And the disease is progressive from onset to death over a course of about 15 to 25 years, as shown in this graph here. Percent survival from the time of disease onset is shown at the 50% survival rate is about at age 18, or age 18 years after disease onset. The causative mutation is a CAG expansion in the gene Huntington. This was discovered in 1993, and this slide is sort of important for understanding much of what I will be talking about later. The Huntington gene is on chromosome 4p16.3. It's really a long gene with 67 exons, and the mutation is an excessively long track of the trinucleotide repeat CAG. So normal individuals have a normal length repeat, and I'll show what that is in a moment, and encode a protein that contains a normal length track of polyglutamine amino acids. The mutant repeat has too many CAG triplets encoding an excessively long track of polyglutamine, which results in the pathogenic process that leads to the disease. The basic genetics of Huntington's disease begins with the fact that it's autosomal dominant, that with a few slight caveats, you have a 50% chance of developing the disease if a parent has it. And if your parent does not have it, neither parent has it, you will not get the disease. The repeat length is important. If you have fewer than 27 triplets, that's the normal range, and you won't develop HD. 27 to 34 triplets is considered an intermediate length repeat. You won't develop HD, but there's some instability so that in one generation to the next, the repeat may expand to the disease range. 35 to 39 triplets is the incomplete penetrance range where individuals may develop HD, and if you have 40 or more triplets, you will develop HD if you live sufficiently long. Two other important facts. One is that longer repeat lengths are correlated with earlier disease onset, and that's shown in this graph here. This on the y-axis is a logarithmic scale of age at onset. This is repeat length, And you can see that individuals who have repeats out in the 60 to 70 triplet length will be developing illness when they're teens or children. And that most individuals have repeat lengths in the 40s to 50s, and these individuals have onset in middle age with substantial variability. The second aspect that I won't discuss in any length is that there is a tendency of the repeat length to increase in subsequent generations, so that in subsequent generations the disease often becomes earlier in onset age and more severe. HD is found throughout the world. It's not restricted to any one ethnic group, though the prevalence does vary regionally. It's about a prevalence rate of about 1 in 10,000 in the United States. There is no treatment to stop the progression of HD. This is a press release from a study of a drug called tominersen, which actually is a small bit of oligonucleotide that was infused in the cerebral spinal fluid to knock down Huntington gene expression, and it was really thought this might be an answer to the disease. And unfortunately, the study was stopped midstream because not only was it not helpful, but people getting the drug were actually doing a little bit worse than people who were on placebo in a dose-response sort of way. So the field is still quaking from this result, meaning that the search for Huntington's treatment remains ongoing. Though I should say that even though there's not treatment to stop the disease, there's still many ways the disorder can be treated to help individuals and their families, such as treating the psychiatric symptoms, which is why this disease is in a psychiatric department for us, and also in helping the family learn and the individuals learn how they can best manage with the symptoms as they evolve. I returned to the Baltimore or Johns Hopkins Huntington Disease Center, which was founded about 1980. There have only been 2 directors. One was Susan Folstein until 1993, and the other has been Chris Ross since 1993. Currently, under the direction of Dr. Ross with the clinical director, Dr. Ji Bang, there are 3 major lines of research. One is to look for quantifiable biomarkers of disease progression. The second is using multiple approaches, cell and animal models, molecular and proteomic techniques, to search for new therapeutic targets. Some of this work is also done by Dr. Wenshen Duan in her lab, and participation and leadership in a variety of clinical trials. When the HD mutation was discovered in 1993, we tested all the individuals in our clinic to see if they had the mutation. It's a pretty simple PCR test. And most had the mutation, but not everybody. And we ended up with a handful of families that had progressive familial movement disorders of unknown cause. So our question was, could we find the causative mutations in any of these families? This is the person that we initially focused on. Her family noticed that she began to have clumsiness and limb and trunk jerks at the age of 26. She first came to clinical attention at age 28 with symptoms of dysphagia, irritability, apathy, and a 20-pound weight loss. She showed on exam abnormal eye movements, some chorea, dysarthria, mild increase in tone, and awkward gait and bradykinesia, and her mini-mental state exam was 26. The diagnosis was made of Huntington's disease by an experienced HD clinician, probably either Dr. Carol Pizer or Dr. Neil Rainin. At age 28 to 35, these symptoms began to increase, and she had a progressive decline in all spheres, so that by age 35, her mini-mental score was 7, but also at that time, the HD mutation had been discovered, and it was found that she did not have the mutation. At age 37, when I first saw her, her progression had continued. She was living in a nursing home. She was dependent for all activities of daily living. She was alert, had one-word answers at best with a mini-mental score of 2. She was rigid and hyperreflexic with a Babinski response, not much chorea. Her strength and sensation were normal, but she was unable to walk, and she passed away shortly thereafter. Based on our findings in her and then subsequent visit to track down her family in the southeastern United States, we were able to discover first that the familial disease was caused by some sort of repeat expansion, and later we were able to clone, this is work by Susan Holmes, we're able to clone exactly where the mutation was and identify the gene. This work at the time took a long number of months. Now it could probably be done by a high school student going from here to here in probably 2 days because of the power of genetic methods and the existence of the human genome sequence. So the HDL2 mutation, it turns out, is not a CAG repeat, at least in the gene in which we found it. It is a CTG repeat expansion in the gene junctophyllin 3 located on chromosome 16. It's a bit confusing, and this has continued to torment us for 25 years because the repeat is here in the CTG orientation, but it's not this, it's in a variably spliced exon, which means that when all the other exons are put together to encode the junctophyllin 3 protein, that exon is missing, so the repeat is not part of the normal protein. It's not part of even the normal RNA transcript. There are some splice variants in which the repeat is included in a transcript, but we now have reason to believe these are fairly minor and probably don't contribute much to disease pathogenesis. But what was interesting is that we found, Susan Holmes found this immediately, on the antisense strand, so the DNA going this way, there was a cryptic gene that was not annotated in the genetic databases in which the repeat is in the CAG orientation, which is to flip the inverse complement, and it encodes polyglutamine. We'll come back to this because it's an important point in thinking about this disease. So the first point I want to make is that we have a case where HD and HDL2 are remarkably similar even though their mutations are different. This is an example of genetic heterogeneity where different mutations lead to the same phenotype. And I just want to go through the proof that we looked at to determine how similar these diseases were. First, or actually eventually, we were able to put together a series of HD and matched HDL2 cases by taking advantage of patient populations in South Africa where HDL2 is more common than anywhere else in the world. This was work led by David Anderson, one of the 3 movement disorder neurologists in South Africa at the time. It's now down to 2 because he moved to Scotland. And Dr. Amanda Kraus, who's one of the lead geneticists in South Africa. So they compared a series of 15 HDL2 patients to 13 HD patients that were matched on the length of the repeat, the age of disease onset, the age of diagnosis, their gender and ethnicity, and their initial manifesting symptom. And what they found was that the phenotype was nearly identical. We'd speculated this beforehand, but this was really definitive proof. They found on all domains that were measured by the standard Huntington's disease symptom rating scale, motor abnormalities, function, cognition, and behavioral, meaning psychiatric symptoms, were indistinguishable between the 2 disorders. And in fact, experts were sent tapes of HD and HDL2 patients and were unable to distinguish who had which disease. And Dave Anderson and Amanda found that the progression of the disease is similar. So on this axis is the total motor score, a rating of symptoms, and on this axis is the duration of disease. And though we don't have tracking of a single patient over time, if you look at patients at different stages of disease duration, you can see in both HD in black markers and HDL2 in the diamond-shaped markers that disease gets worse over time, and the rate of progression appears to be similar between the 2 diseases. We also examined MRI scans, and here again, HD and HDL2 MRI scans are essentially indistinguishable. In a normal individual, these are the rather small lateral ventricles with the caudate nucleus kind of indenting into the lateral nucleus. And in Huntington's disease and Huntington's disease-like 2, those caudate nuclei are gone, so is much of the rest of the basal ganglia, which is located here, and it's largely atrophied in both HD and HDL2. Meanwhile, the cerebellum is normal, as is the brainstem. Dave Anderson later quantitated the structural findings in HD and HDL2 and confirmed what we had observed clinically, that is, that the volume loss is similar in the two disorders in similar regions, with the odd exception that the thalamus seems to be more atrophied in HDL2 than HD. As one might expect from imaging findings, the pathology is also nearly identical between the two diseases. In this case, 5 HDL2 brains were compared to match HD brains, and in our group, and both had mild to severe cortical atrophy, marked striatal atrophy, reduced white matter volume, though it was well myelinated. And once again, when we sent out pathologic samples to experts in the field, they could not distinguish HD from HDL2. Microscopically, the diseases also look remarkably similar. This slide here shows the presence of these protein aggregates that had been found a long time ago in Huntington's disease that stain with antibodies either to ubiquitin, which marks protein aggregation, or to an antibody specific for long tracks of polyglutamine. The same kind of protein aggregates or inclusions were found in HDL2 brain samples, and even if you look at them by electron microscopy, they even look the same. They have this loose fibrillary material, looks like this. Again, indistinguishable one from the other. There are some slight differences in where the inclusions are located in the brain, but the similarities far outweigh the differences. There's also a similarity in the relationship of the repeat length to disease in HD and HDL2. As I mentioned before, the threshold for disease is about 40 triplets for each disease, which is well defined in Huntington's disease. It's a little harder to define in HDL2 because the sample size is much smaller, with perhaps 100 patients being described around the world. Also, like HD, the repeat length in HDL2 is inversely correlated with age of onset. Here again is age of onset, and here's repeat length, and you can see a correlation. Longer repeat length means earlier age of onset. The slope was actually remarkably similar to HD. We've subsequently repeated this with individuals from South Africa with pretty much the same result. There's similarity even at the protein level, where there's a large overlap in the Huntington's disease and the Huntington's disease-like 2 proteins that are dysregulated. This is work that's ongoing by Steve Dahl in my laboratory, where he looked at the fraction of proteins that are collected in the nucleus that are not soluble, so the particulate matter essentially, and then he took a look at what those proteins were using mass spectrometry. And what he found was that there was a substantial overlap in the proteins that were enriched in this type of protein fragment from the nucleus of cells that were the aggregated matter, and also an overlap in the downregulated proteins that were depleted in this region compared to controls. So we don't know exactly what this means yet. Steve has some ideas, but it again suggests that there are substantial similarities between the disease, though also some differences at the protein level. So, HD and HDL2 arise from these similar mutations, actually similar mutations that are but in different genes and different orientations, and they have similar clinical, imaging, pathological, genetic, and proteomic phenotypes. How is this type of genetic heterogeneity, that is, similar diseases arising from different genetic mutations, relevant to common psychiatric disorders? Well, there's an example of the application of genetic heterogeneity to schizophrenia and related disorders. This comes from work done by the Schema Consortium, which recently established a series of genes that have mutations that each greatly increase the risk of developing schizophrenia, risks that up to as much as 50 times the background rate. This work is ongoing, and they've recently revised their list, but the basic principle remains. We're exploring some of these mutations with a hypothesis that comparing the neurobiologic impact of these different mutations will reveal pathogenic pathways that may serve as targets for therapeutic agents. Which pathways are common and which are not common among these affected genes that actually have very different functions. This is being done by work in Pam Lee's lab, in Chris Ross's lab, and by Hannah Jaropelid in my lab, looking at the loss of function of these genes and the role of specific mutations using various forms of cell models, particularly engineered stem cell models and genetically engineered mouse models, as well as the idea that marmoset models may also be valuable. And we anticipate that the same approach will be of value in other disorders, in particular mood disorders, which have a substantial degree of genetic overlap with schizophrenia and schizoaffective disorder. So the second point I want to make is that mutations are complex, and HDL2 is a perfect example. Here's the diagram of HDL2 pathogenesis that was generated by Pam Lee. And I want to go over it part by part. First, there is a component of disease pathogenesis that we suspect may be from the antisense part of this gene down here, which we depict down here, making a protein that contains too many polyglutamines, leading to neurotoxicity. And this is supported by both the phenotypic similarity of HDL2 to HD, the presence of polyglutamine containing aggregates in HDL2, and a mouse model in which the junctophyllin 3 antisense strand is expressed and which develops a motor and cognitive phenotype. But that's not the only way that we think that the mutation causes disease. The second aspect is loss of functional junctophyllin 3 protein. And here we have evidence that there is a decreased expression of junctophyllin 3 transcripts with a loss of junctophyllin 3 protein. And junctophyllin 3, it turns out, is important in regulating how calcium is released when cells are signaled, when electrical signals come into a cell. And so we suspect that abnormalities of calcium influx leads to toxicity. There's evidence for this based on a mouse model in which junctophyllin 3 is lost, and that mouse develops abnormal behavior, and that mouse also develops neurons with abnormal electrophysiological properties. The third mechanism is that there may be toxicity caused by the RNA transcript without even needing to encode for protein. And in this case, we have evidence that in the brain of people with HDL2, the RNA from junctophyllin 3 forms clumps like this. These clumps begin to grab onto other proteins that are floating around and prevent those proteins from functioning, and that loss of function from proteins that are brought into the RNA foci, they're called, they're really aggregates of RNA, lead to toxicity. And here we know that this mechanism exists in other diseases, and we found the RNA inclusions, and we have some in vitro cell models showing how this mechanism can be toxic to neuronal-like cells. So, mutations can be complicated, and we suspect that this complexity is likely to play out in more common psychiatric disorders. A single mutation may alter multiple cell processes, or different mutations in the same gene may have different pathogenic effects. The experiments in the Division of Neurobiology with the schema consortium genes will be testing these hypotheses. The last point I want to make is about genetic diversity. It turns out that HDL2 is a disease of those with African ancestry. It's only been found so far in individuals who are definite or of likely African descent. And if you look in populations, we find that HDL2 in individuals of African descent is as common in South Africa, or almost as common in South Africa as HD, and in our center represented about 15% of HD-like cases, so that it is an important cause of disease in individuals with African descent. This is pretty interesting. So this slide is a picture in diagrammatic form of the slave trade from about 1500 to about 1900, from Africa to South Africa, the Caribbean, and to North America. It's simplified and the like, but it makes a point. What we have found, particularly the group in South Africa, is that HDL2 cases are found scattered around South Africa, around Africa, most prominently in South Africa, but that's because the infrastructure for detecting genetic diseases is much stronger in South Africa than most of the rest of Africa, and that the dispersion of HDL2 matches that of the slave trade. South Africa, high pop percentage in Caribbean and in the United States, as well as some cases in Mexico, which leads us to predictions that this area of Africa, which has not been studied systematically for HDL2 or other neurodegenerative disorders, is likely to have a relatively high prevalence of HDL2. And further, this is an example of how genetic diversity in Africa is really a unique opportunity. This slide this shows a bunch of different populations that were studied by one of a number of different genetic studies in Africa, and this slide is a depiction of the various, an example of the genetic diversity among these populations, which is markedly greater than in the rest of the world. This suggests that additional rare mutations of high impact remain to be discovered, not just in HD-like disorders, but in other psychiatric and related conditions, and that looking at the pattern of genetic variability that contributes in aggregate to disease risk is likely to be different in African populations and may be quite informative in understanding how complex diseases that arise not from a single mutation but from a number of small variables, very small variations in gene expression occur. So just to summarize, HDL2 is a neurodegenerative disorder strikingly similar to HD. The HDL2 CTG repeat expansion mutation is located in a different gene, in a different orientation than the HD CAG repeat expansion, yet leads to a phenotype that is almost identical. HDL2 is found only in individuals with African descent, at least so far, predicting that there'll be areas of relatively high prevalence that have, where individuals have yet to be diagnosed, particularly in West Africa, and is an example of the value of looking at genetic diversity when studying largely genetic diseases. So to end with the words of Bateson, if I may throw out a word of counsel to beginners, it is treasure your exceptions. When there are none, the work gets so dull that no one cares to carry it out further. Keep them always uncovered and in sight. Exceptions are like the rough brickwork of a growing building, which tells that there is more to come and shows where the next construction is to be. HDL2 is our rare exception that we indeed treasure. So again, my thanks to the many individuals who have been part of this project, the funders, and of course to our patients.
Thank you, Dr. Margolis. Very, very interesting. I guess I'll just first make the point you probably are well aware of. You probably know that our head of the Department of Genetic Medicine, who's been with us maybe a year or two now, Ambrose Wankum, is a native of Cameroon in West Africa and has been very involved in studying African genetic diversity.
And in fact, he was part of our consortium that applied to Chen Zuckerberg.
Oh, great. Okay.
No Facebook for us anymore.
That's great. But maybe more relevant for folks here, I wonder if you could say a few words about schema. So schema is a consortium looking for rare mutations that have a large effect in schizophrenia. How big a role do these rare mutations have in causing schizophrenia?
Yeah, I mentioned that last time, and I did grand rounds in February. These genes have a mutation in some of the genes that were discovered by this consortium can increase the risk of schizophrenia close to 30 to 50 times the background rate, which means the background rate is about 1%, that a mutation in one of these genes means you have about a 50% chance, at least at the high level, of developing the disease. And some of the other genes are more like 20 or 30% increased risk, as compared to the minor variations that are detected in GWAS studies, where the increased risk is about 1% at most.
I guess I'm tempted to go on and on, but I'll give other people a chance.
It's a great study.
Dr. Sedlak.
Thank you. I wonder if you could offer some perspectives on gene therapy for psychiatric disorders and HD. One of the challenges for psychiatric disorders are genes may affect brain development, but we're seeing individuals in their brain has largely completed its development. Do we know much about the HD genes if you can relay their gene function to output in the models? Would you still see the phenotype or they're more specific for the onset?
So just repeat the question. The question is about gene therapy and
Gene therapy, both in more common psychiatric disorders that tend to have neurodevelopmental factors, and of course we're nowhere close to anything like that, and it is, I think, a major concern about what the value will be in a disease where neurodevelopment has already occurred. But I think, that being said, there is also reason to be somewhat optimistic that some processes are ongoing and that beneficial impact can occur even after neurodevelopment has ended. In Huntington's disease, and Huntington's disease like 2 eventually, the situation is a little easier to define. There's been a huge amount of work in Huntington's disease to try and figure out when the disease first manifests and when the earliest changes occur, so when one might need to intervene. And I think the general consensus, and Chris Ross can comment further, is that one wants to intervene ideally before the disease begins to show symptoms, maybe by 10 or 20 years, but doesn't have to be done immediately at childhood in order to have a major effect. So gene therapy in a variety of forms is, in fact, actively being pursued in Huntington's disease, mostly not to change the gene, but to knock down the expression of the mutant alleles.
Other folks. Dr. Ross.
Yeah, actually, I wanted to comment on the issue of genetic diversity and heterogeneity, because I think what's coming out of the schema consortium is that there are these rare genes that have very high elevation of risk for schizophrenia. The work in bipolar disorder is not as far along, but there are also high-risk genes for bipolar disorder. Some of them are different, but there is at least one example of a gene which is the same, and I think it kind of supports the concept of a spectrum between bipolar disorder and schizophrenia, the schizoaffective disorder concept. And furthermore, some of those genes that have very rare but high effect mutations are also coming up in the GWAS studies, so they are clues to the more common risk factors for both bipolar disorder and schizophrenia. So I think that's just kind of another example of how this kind of thinking can help us understand the heterogeneity of the disorders we see in the clinic.
Mm-hmm. Thank you. Thank you. Dr. Tobolowsky.
I was curious, so we learned a little bit more about the lymphatic system and the role of abnormal proteins. I'm curious if there's been any connections here tied to like sleep and age of onset for Huntington's disease or HLP2.
I think there's some very early work being done by Wenshen Duan here in conjunction with the neuroimaging group to look at lymphatics system with the idea that impairment may be of lymph flow through the brain may be a factor that leads to abnormal protein deposition. And it's certainly something that we're interested in exploring in schizophrenia based on the finding by Dr. Nusafora up there of a certain subset of patients with schizophrenia that have abnormal aggregates of protein. And so one wonders, could there be abnormal lymphatic flow Could that relate to sleep? Maybe. That's beyond where the research is now, but it's an important direction of research.
Other folks? Yeah, go ahead. Jess.
The work about Huntington and Free, have there been any rescue experiments that kind of look to see what contribution of phenotypes due to that defect versus the iron?
Yeah, well, in a way. The question is, have there been rescue experiments? And so, one of the issues we've been seeking to tease apart is how much of the phenotype, particularly in mouse models, is from loss of function or from gain of function from a polyglutamine made from the reverse strand or from some RNA toxicity. And we know that a mouse with missing copies of junctophyllin 3 is abnormal. We know that a mouse that only has a mutation that expresses in the antisense strand is abnormal. And so one would think that that suggests that there's a combination of the two factors. Trying to do rescues, I think, is downstream for us as we're developing new and better mouse models, but it is trying to disentangle all of this it turns out to be kind of confusing. And some of our experiments have not been successful, but it's an important point.
Other folks? Go ahead, Dr. Kim.
The evidence, unfortunately, has been very mixed. On the one hand, aggregates in HDL2 stain with antibodies that are specific for polyglutamine. And so one say, aha, of course there must be polyglutamine, that must be the cause of the disease. But when we look on Western blots, for instance, we can't detect that protein for reasons which we do not understand. And so it remains uncertain, and a point that's bothered me for 20 years.
Well, just in the interest of time, since we just spent out of time, I want to ask a different question, which is that, of course, one of the goals of genetics is, one goal might be to get directly to a therapy, like a gene therapy, as you talked about, and another big goal has been that to use genetics to get to understanding pathophysiology, what's happening in the brain, which leads me to the question, I suppose you could answer, we could ask it for HD, but for schizophrenia, to what extent has identifying those genes told us anything about what those genes are doing together to cause schizophrenia in the brain?
Ask that question in 5 years. But I mean, the course is there. I mean, the discovery of the genes, the neurobiologic methods to study them are all now exist. And in fact, it's one of the reasons we began in our group studying Huntington's disease, was to build up a repertoire of tools and approaches that can be applied. Now the genes have been discovered and they can be disentangled. And I think there's real reason to be optimistic about that.
Mm-hmm, mm-hmm. Okay, thanks very much for a great round.